I opened things up a little bit early. We'll get started in a few minutes. All right, guys, let's go ahead and get started. So let's see. Let me make sure I'm sharing the right screen here. Yep. All right, so news and reminders. The class is wrapping up quite rapidly. So just as a reminder, there is no class on Thursday. Thursday is Thanksgiving. That we have next week left and then the week after. And really what it comes down to is there's only 2.5 lectures left. By that I mean we have the 2 lectures next week. One is going to be on translation. The other one is going to be on DNA replication. And then the last Tuesday is going to be sort of dedicated to kind of catching up. I think the translation lecture, the material is going to have to kind of, that's going to bleed into the DNA replication material, which is going to bleed into Tuesday, I think. But there won't be that much stuff. And then the rest of Tuesday will be dedicated for you guys to ask me whatever questions that you might still have lingering. I could talk about the exam a little bit as well. So not a lot left. I haven't graded the weekly quiz that I gave out. There won't be a weekly quiz for this week. So again, things are wrapping up. Ooh, one thing. Let me almost forgot. Keep working on those journal article questions. So we'll talk about some of the journal article stuff today again. I think we got to almost everything. There's a few things I still would like to touch upon that we'll get to eventually, but you should have pretty much all the tools necessary to answer all the questions within that journal article now. Okay, good. I think that's all the news and the reminders that I have. Before I sort of move into my little introduction into the material that I'm going to send you guys out to discuss, do you have any questions, any concerns about the logistics of the course or due dates or what have you? You can pop it in the chat box, unmute yourself and ask if you have it. Right, well, so what the sort of jumping off point for today is what we left off with last Thursday, which is that if you were to look at the regulatory sequences, so the sequences that control when and in what cells a particular gene is going to be expressed, you'll see that it's identical in all cells. And the reason for that is we as a multicellular organism started as a single-celled embryo, a zygote. And then that zygote just simply went through mitosis over and over and over again to give rise to you and I. But more complicated than that is that different cell types started making different activators, and those activators will bind to the regulatory sequences and turn on specific genes. So even though the regulatory sequences of all genes are the same in all cells, the activators that are being expressed in those cells are different. Now, the ultimate question we ended with is, how does that happen? If we started as a single cell, and that cell just goes through mitosis over and over and over again, excuse me, how is it that the different cells started making different activators? Where did those asymmetries begin, and how did those asymmetries begin? So with that said, the group of questions that you are going to be working on for our first, can you post the lectures so far, please? Oh, the lecture recordings. Carla, I did that last night. So I don't know when the last time you checked, but it should be all updated by now. When was the last time you checked, Carla?
I checked last night, but it was kind of early-ish.
Oh, okay. Then I think, yeah, I think it was sometime after that I updated it. So
Oh,
check again.
okay.
If it's still not updated, let me know and I'll get that. But I'm pretty sure I got to that like last night. I don't know exactly what time.
Oh, okay. Thank you.
Yep. Okay, guys. So what I'd like you to work on for this first session is questions 1 through 14. So talk about cytoplasmic determinants and inductive signaling, and then talk a little bit more specifically about retinoic acid. So 1 through 14. Let me get this. Copy that link. Toss that link over here. Let's see how I'll recreate my breakout rooms. Let's see. I'm gonna Tulane. We'll take a look. It looks usually Tin gets a little bit later, so we'll give you a few minutes, and then if Tin doesn't show up, I'll toss you in group 2. But otherwise, here you guys go. Hannah, what group are you in?
Group 2.
Oh, you're in group. Okay. All right. Yeah. Okay. Yeah.
So should I join the room or should I wait?
Why don't you go into that room, and then if within the next few minutes, if he doesn't show up, I'll move you. Or you know what? I'll tell you what. Yeah, go into that room because I can't do anything until you go into that room,
Okay.
and then I'll move you into 2 immediately, and then we'll see what we'll adjust by there. So go ahead and go into that room, and then I'll move you. All right, guys. So I've got just 2 clicker questions here for you. So let me share my screen. There. Share. And okay, so let's ask number 1 first. Give another 15 seconds. I think there's just 1 more of you left. There you are. All right. D, all of the above. Yes, good. So these cytoplasmic determinants, right? These are molecules. Let's do this. Let's take advantage of this point here. So these cytoplasmic molecule, they are molecules that have been loaded into the egg's cytoplasm by the mother. So that's how so all of these in the zygote, right? When you are fertilized when the egg is fertilized by the sperm, giving rise to that first single-celled embryo, all of these various factors here that asymmetrically divide in that cell, that or portion themselves out in that cell, all come from your mom. They were loaded into that egg even before it was ever fertilized by a sperm. So you've got all of these proteins that come from mom. Now, the purpose of those proteins are to do a couple things. Well, ultimately, the purpose is to turn on genes necessary to give rise to proper development. Now, how can we do that? How can proteins turn genes on? Well, the most obvious that you guys should know is, of course, activators. these proteins that bind to enhancers that turn on genes. But don't forget that oftentimes, especially for eukaryotic cells, that what else needs to occur is the chromatin needs to open up, and some of those N-terminal tails need to be modified to attract activators, to attract other proteins necessary for genes to be turned on. And so any one of those molecules can be a potential cytoplasmic determinant. Molecules that bind to DNA, molecules that modify chromatin, molecules that modify the N-terminal tails of histones. All of those are going to be represented here in these cytoplasmic determinants, okay? All right, next question before we kind of open the floor up a little bit is number 2 here. Give you another 20 seconds. Okay, that's it, I think. Excellent, good, 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 good. 2 of the above. So let's talk about this just a little bit here. So I'm going to stop my share. Come back here. And I'm going to do this. Nope, that's not what I wanted to do. Let's try it again. One moment. Why isn't that working? That. Aha. We have some technical difficulties, but fixed. So the retinoic acid receptor protein is a pretty complex protein. It has a region, okay, the first and most important region is being a retinoic acid receptor, it needs to bind retinoic acid. So it has a region here that binds retinoic acid, and we call that the ligand binding domain. Binding domain. Another word for signal is a ligand. So either the signal binds to the receptor, or you could say the ligand binds to the receptor. And then we've got another part here, right? Boop, 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 boop. Let's call that this part here. This is the activation domain. This is going to be the domain that attracts other proteins, that sticks to other proteins. Now, this activation domain, as the majority of you got right, this is what is going to recruit either the PRC2 complex or the trithorax complex. Now, which one it recruits will depend upon whether retinoic acid is actually bound to the receptor or not. If the retinoic acid is bound to the receptor, it changes the conformation of this region such that it binds to the trithorax complex. If retinoic acid is not bound, then the conformation of this changes such that it binds to PRC2. And finally, the other part that we need here is the DNA binding domain. So this is going to be the region of the retinoic acid receptor that allows it to bind to the retinoic acid receptor element. Now, the neat thing about the retinoic acid receptor is that it can bind to this retinoic acid receptor element in the presence and absence of retinoic acid. So it just sits there. It sits there regardless of whether or not retinoic acid is around. And then what happens is, depending upon whether retinoic acid is around, will determine the state of this activation domain and whether it's recruiting PRC2 or whether it's recruiting trithorax. Okay, so it's some complex protein. Okay, let me give my face back. There I am. And then let's close this up here. Whoops. All right, and then we'll go here. So basically we've got 1 through 14. Does anybody need any further clarification in questions 1 through 14? You can unmute yourself. You can ask it via the chat box there. Whatever you want. So is 12, so let me see 12. 12, what does the DNA binding domain of the retinoic acid receptor bind to? Yes, that is, no, not trithorax. So for the answer to number 12, what does the DNA binding domain bind to? That's going to be the retinoic acid receptor element. That's going to be the sequence of DNA, the enhancer, the regulatory DNA, right? We can call it all kinds of different things, but it's going to be a sequence of DNA. Okay, good. Okay, so yes. All right. Any other questions? I'll just wait another 30 seconds or so just in case you guys are typing something. In the meantime, I want to take you to this. So developmental biology, guys, is a course when you transfer, or if you transfer to a 4-year institution and you have a couple higher level biology courses that you need to take and you're not sure what, I highly, highly, highly recommend taking a developmental biology course. And if it's offered, take a developmental biology lab. Because in these labs, you get to do some really neat experiments. I don't know, it's been years and years and years since I've been in a developmental biology lab. So I don't know how they may or may not have changed over these years. But some classical developmental biology labs will incorporate these demonstration of inductive signals. So there's these famous tissues. As an embryo is developing, there's these famous tissues called organizing centers or inductive centers or something like that. So for limbs, as a limb is developing, there's a little patch of tissue here, meaning a group of cells that's called the zone of polarizing activity. What's special is the cells in that ZPA there are secreting inductive signals. Secreting the signals necessary to turn on developmental pathways in adjacent cells that makes them become and develop into a limb. And now this zone of polarizing activity, to get it set up, to get it to become the zone of polarizing activity, is dependent on retinoic acid. And to demonstrate this, so they have this hypothesis. We hypothesize that retinoic acid is an inductive signal necessary for the induction of the zone of polarizing activity, which in itself is responsible for inducing the limb. So what do? If that's true, then if they take a little glass bead or a little sort of a porous bead that has been soaked in retinoic acid, and they implant that in a different area of where a limb bud usually forms. So Here we've got a limb bud naturally forming because there's already a naturally occurring zone of polymerizing activity right here. As that's occurring, they pop this retinoic acid bead right here. The idea being it's going to cause another zone of polarizing activity, ZPA. And sure enough, when they implant that bead there and then let this limb bud begin to develop, they show that they get a mirror duplication of elements of that limb. This would be an experiment that you might do in a developmental biology lab. Furthermore, if you haven't watched that video, you really should. Speymann's organizer. So during early development, right before a process called gastrulation, there is a piece of tissue called the dorsal lip. We're not going to go into the anatomy of an early embryo. That's beyond the scope of this course. But what's important to understand is there's signals that emanate from this dorsal lip, this Spemann organizer, that convert all of this tissue up here into a dorsal fate. So that would be the back. And they demonstrated this by taking and removing this tissue from an embryo and inserting that tissue into another embryo that had a normal Speymann's organizer, so that was operating normally, into the ventral region. So now this embryo had essentially two dorsal lips, two Spemann organizers, both sending out signals, both inducing the back region here, inducing the back region here, the dorsal region here, and sure enough, you get an embryo that has been that's essentially duplicated body axes. So it's some pretty cool stuff. That's another experiment that you might do in a developmental biology lab. All right. So I just wanted to kind of take that as an opportunity to advertise the fact that you guys should take a developmental genetics course someday. Which means, let's see, we did questions number 1 through 14. Let's now do questions 15 through 22. 15 through 22. And open all rooms. There you go. Well, you guys took a little bit longer to get back after I closed the rooms. Did you need more time to work on these questions? How are you guys doing? I'm sorry. You guys are you good for going into the clicker questions? Yeah?
I don't think so because we're working on the
You're still working on it? Okay,
yeah.
okay. Yeah, you guys took suspiciously long time to come back from the breakout room. So why don't you tell me which number were you on? Were you working on somebody in your group? You could put it in a chat box or unmute yourself, whatever.
Actually, we just start working on it, so it's gonna be a 19 question. The question 19.
Oh, okay. All right. Awesome. Okay, so I'm going to send you guys back out. I'm sorry, I did that a little bit prematurely. So here you go. Okay, guys, let's go ahead and ask. I think I just have 2 clicker questions. I got to rearrange some stuff. One moment. All right, here we go. Let's see. We are at, whoops. There we go. Okay, so number 3. Give you another 15 seconds. All right, I think that's it. And, ooh, all right. So the epigenetic mark that's associated with the expression of HOXB1 is going to be B. So 50% of you got that one. So if we take a look at that, let's go here. Let's go, where is that? Here. Right, so this is what we have. H3K4 is trimethylated. H3K27 is completely demethylated. And it's only then that we get activation of HOXB1. So that is the epigenetic mark that sort of indicates that or causes the HOXB1 to be expressed. All right, let's do number 4. One second, hang on. I think I need to fix this. Wait a minute. Presence of retinoic acid this should be D. Sorry, you could change your answers. All right. Okay. And the answer is, so in the journal article demonstrates that the presence of retinoic acid, more H3K27, is demethylated in the region around the HOXB. The answer, it's true. If we take a look, let's take a look at that experiment. Let's see, where is it? Over here. So if we take a look at that experiment, right? Here is, right, this is the chip experiment. It's showing us how much of this DNA here is bound by trimethylated K27 in the absence and the presence of retinoic acid. And what they show is that in the absence of retinoic acid, quite a bit of that K27 is trimethylated. In the presence of retinoic acid, most of it is demethylated. And that's exactly what that question was asking, is did it show, let's see, that figure 3b in the journal art demonstrates that in the presence of retinoic acid, more H3K27 is demethylated. And that's indeed what they showed. In the presence of retinoic acid, we have more demethylated K27ME3, right? In this case here, it's all trimethylated. In this case here, it is not. All right, last question. Let's see here. Number 5. All of you got that? All right. Okay. I thought this. So this is an important, and we'll talk about this, but this is false. So this figure is not showing us that UTX is necessary for the methylation of H3K27. Okay. So let's take a look at why that's the case. Let's see, I'm going to stop my share. And let's see, actually, let's go ahead and share this. Yeah. So what this figure shows, let's go here. What this figure shows are just correlations that in the presence of retinoic acid, we get UTX localizes here. We get that there is less trimethylation of K27 here. We get less EZH2 localizing here. We get more trimethylation here. That's it. That's the correlation, that it's just correlations that are being made, right? This experiment is not telling us that UTX, right? If this experiment was let's think about it this way. Let's think about it this way. If this experiment was telling us that UTX is what is demethylating K27ME3, if that experiment was telling us that, then by the same logic, it must also be telling us that UTX is also causing the trimethylation of K4, right? But it's not. It's not telling us that. It's only telling us a series of correlations. It's only telling us that in the presence of retinoic acid, this UTX is here, this trimethylation occurs here, EZH2 drops off of here, and K4 becomes trimethylated. It's just telling us that all of those things are occurring in the presence of retinoic acid. It is not saying that one is necessary for the other is necessary for the other. Okay, because it's quite possible, for example, so if we do this, let's look at an alternative hypothesis here. It is quite possible, one moment guys, I gotta stop my share. So if we have our situation here, here's HOXB1. This is the retinoic acid receptor that's sitting at the retinoic acid receptor element. And here is our H3K27 trimethylated thing. And so now when retinoic acid binds, right, what we have is that attracts JMJD, that attracts UTX. So here comes UTX. And it causes H3K27ME3, right, to become H3K27. That. But this experiment doesn't tell us that retinoic acid receptor bringing in UTX is what causes this. It's quite possible that retinoic acid receptor brings in another molecule that we don't know that is responsible for this, right? This hypothesis will also give rise to the same data that we see in figure 3b. Retinoic acid receptor recruits UTX. Retinoic acid receptor recruits protein X that we don't know yet. And it's protein X that demethylates H3K27ME3, right? This particular possibility would give us the exact same results that we see in figure 3b. Right? So it's really important to understand that. What kind of experiment needs to be done in order to prove that UTX is what is causing this? And this was not a rhetorical question. I want somebody to volunteer to answer, what experiment do the researchers need to do to prove that UTX is indeed the cause of the demethylation of H3K27ME3? So, anyone want to volunteer an answer? You can type it up. You can unmute yourself. What experiment? I'll give you a hint. It's an experiment that they've already done. So what experiment did they do to prove that H3 or that UTX is the cause of the demethylation? The Western blot proves that it can demethylate. Yep, it can demethylate, but that is in that particular experiment. Let me see if I can bring that up here. Hang on a minute. Where is it? Oh, it's on my other. That experiment was done in vitro and not within cells. So we want to make sure that we look at this in the context of actual cells. Give me 1 second and I'll bring up, let me stop sharing. Oh, I wasn't, okay, 1 minute. Where's my share? Stop sharing. So, if we look at, oh, I gotta get all my chats here. These experiments right here, right? This experiment here demonstrated that if you overexpress UTX or JMJD3, you lead to more demethylation. This experiment here with using that short hairpin UTX shows us that if we get rid of UTX, we have more trimethylation. All right, so these two experiments showed us that not only is UTX and JMJD3 sufficient to cause demethylation, it is necessary for that demethylation to occur. In one experiment, we overexpressed it and saw that we saw more demethylation. And in the other experiment, we removed it and showed that we got less demethylation occurring. So it's experiments like these that demonstrate whether or not something is causing some phenomenon to occur. In experiment 3B, we weren't doing that. We were simply showing that, okay, this is a region of DNA that we're interested in, and in the absence or presence of retinoic acid, these are the things that occur there. Now, what causes what? Well, we need to do some other experiments to tease that out. And here are some other experiments they did to tease that out. All right. Does anybody have any other questions? Well, it is 1:08. Class ends in about 7 minutes. Okay, yes, exactly. So, Carla, the answer then from A to E is no, 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 no. Exactly. Yep. Let's see. So let me do this. Let me stop my share, new share. Here. All right. So what I have left, but we're not going to have enough time to go through, is this here. Well, what time do we have? 1:10. We got 5 minutes. Let's save this for Tuesday, the strategies of regulatory sequences. And for now, does anybody have any other questions regarding this material? You can unmute yourself, put yourself in the chat box. All right, I think I'm going to let you guys go. Get out 5 minutes early. Go prepare for the weekend. Be safe. And I will see you on Tuesday. See you, Carla. You too, New. Happy Turkey Day to you, Wayne.